Bismillahirrahmanirrahim. Assalamu alaikum dear students. We are here for the 13th lecture of Introduction to Biochemistry, and the topic is physical and chemical properties of various classes of carbohydrates. In this lecture we will discuss about the carbohydrates briefly, then we will discuss about the physical and chemical properties of monosaccharides, disaccharides, oligosaccharides and polysaccharides. The mutor rotation is defined as the change in the specific optical rotation representing the interconversion of alpha and beta forms of D-glucose to an equilibrium mixture. For example, when the D-glucose is crystallized at room temperature and a fresh solution is prepared, its specific rotation of polarized light is +112, but after 12 to 18 hours it changes to +52.5 degree. So this change in rotation with the time is called its mutor rotation. Now here are some chemical properties of the monosaccharides. Some of the important chemical properties of monosaccharides are its furfural formation, enolization or tautomerization, oxidation reduction, osazone formation, glycoside formation or its ester formation. The first one is the furfural formation. When the sugars treated with the strong mineral acids like concentrated sulfuric acid or hydrochloric acid or concentrated nitric acid, they undergo dehydration and loses 3 water molecules, and finally they form a furfural derivatives. This is the basis for following reactions: the Mollis's reaction and the Selivanov reactions. Both of these tests are used for the detection of the carbohydrates, qualitative detection of the carbohydrates. Here's the reaction for the furfural formation. In case of D-glucose, hydroxymethyl furfural is formed when the glucose is treated with a concentrated acid, and this further condensed with the phenolic compounds and form a colored complex, which is a purple ring at the junction. So this is used for the qualitative detection of the carbohydrate. The next is enolization or the tautomerization, and which is the action of the alkalis. When the carbohydrate solution is kept in an alkaline solution for several hours, it undergoes isomerization to form D-fructose and D-mannose. So this results in the formation of a common intermediate which is known as anidiol, and the process of shifting of hydrogen atom from one carbon atom to another to produce anidiol is known as tautomerization. Here you can see the chemical process of the formation of anidiols. When the glucose is kept in alkaline solution for several hours, it undergoes isomerization to form its form D-fructose and D-mannose, and this results in the formation of a common intermediate which is present in the middle and known as anidiol. And this process of shifting of one hydrogen atom of one carbon atom to another to produce anidiol is known as tautomerization. So anidiols are good reducing agents and form the basis of reducing property of the sugars. Two tests are employed for the qualitative detection of carbohydrates, and which are on the basis of the anidiol formation. The first one is Benedict's test and the second is Fehling tests. Both of these are the qualitative tests for the detection of carbohydrates, in which the red precipitates are formed, and ultimately it depicts the presence or absence of the carbohydrates in the solution. When the alcoholic group, aldehyde or ketone groups which are present in the carbohydrates are oxidized to carboxyl group with certain oxidizing agents, the oxidation may occur and this may brought under mild or vigorous oxidizing conditions. For example, the first condition is with the mild oxidant like BrH2O. In this group, only aldehyde is oxidized to produce gluconic acid or monocarbonic acid, and ketosis do not respond to this reaction. So this reaction is specifically for the detection of aldehyde. If this with the strong oxidizing agent like concentrated HNO3, so both aldehyde or ketone groups are oxidized to yield dicarboxylic acids. The third condition is oxidation with the metal hydroxides. The metal hydroxides like CuOH2 or AgOH oxidize free aldehyde or ketone groups of mutarotation sugars and reduce themselves to lower oxides of the free metals. The second is reduction. So both aldoses and ketoses may be reduced by enzymes to the corresponding polyhydroxy alcohols. The sugar alcohols function mainly as intermediates in the minor pathway of the carbohydrate metabolism. Some of these alcohols are the following: sorbitol, mannitol, dulcitol, ribitol. Some of the alcohols are following. For example, from D-glucose, a D-sorbitol is formed, D-mannose into D-mannitol, D-fructose into D-sorbitol, D-galactose into D-dulcitol, D-ribose into ribitol. So mannitol, it is used as an osmotic diuretic to reduce the cerebral edema. Whereas the sorbitol, it accumulates in the lens of diabetic and produces cataracts, which is a disease condition of eye in the diabetic condition. The next important chemical property of carbohydrate is osazone formation, which is the action of phenylhydrazine. Only reducing sugars give their test positive for their osazone formation. The presence of free carbonyl group, which is aldehyde or a ketone group, in the molecule is essential for the osazone formation. So osazone are yellow crystalline derivatives of reducing sugars with phenylhydrazine and have a characteristic crystal structure which can be used for the identification and characterization of different sugars having closely similar properties. Osazone formed from glucose, fructose and mannose are identical because these are identical in their lower 4 carbon atoms. Here you can see the structure of the glucose, fructose and the mannose, and the last 4 carbon atoms are identical in their are identical. So non-reducing sugars like sucrose cannot form osazone due to the absence of a free carbonyl group. So the osazone crystals of some sugars are glucazone, which is a needle-shaped osazone, maltosazone, sunflower petal-shaped, and lactosazone, power puff of tennis ball-shaped osazone. Here you can see the representation of osazone formation, which is a needle-shaped crystals, power puff or tennis ball-shaped, sunflower petal-shaped osazones, and these can be used for the detection of several types of carbohydrates. Glycosides. In formal terms, a glycoside is any molecule in which a sugar group is bonded through its anomeric carbon to another group via glycosidic bond. This group may be with the carbohydrate linked or with the carbohydrate plus a non-carbohydrate group. If it is condensed with a carbohydrate group, then it is called glycoside. And if it is condensed with the non-carbohydrate group, it is called a glycon. So we can define a glycoside is as when the hemiacetal group of a carbohydrate moiety is condensed with the alcohol or phenol group of another carbohydrate, it is called glycoside. and if it reacts with the non-carbohydrate group, then it is called a glycon. As we know that the bond joining the monosaccharides is called glycosidic or the glycosyl bonds. There are two types of the glycosidic bond. The first one is O glycosidic bond and the second one is N glycosidic bond. As we already discussed that the carbohydrate can react with their anomeric carbon or carbonyl carbon with another carbohydrate or the non-carbohydrate moieties. If the carbohydrate group reacts with the alcohol group, then they form the O-glycosidic bond by the liberation of the water molecule. But if the anomeric carbon of the sugar forms the bond with the nitrogen atom of an amine, the bond is then called an N-glycosidic bond. Glycosides do not reduce Benedict's reagent because the sugar group is masked. They may be hydrolyzed by boiling with the dilute acid so that sugar is free and can then reduce copper. Some glycosides are of medical importance. For example, digitonin, which is a cardiac stimulant, and fluorescein is used to produce renal damage in the experimental animal. Here you can see the O-type glycoside bond and the N-type glycosidic bond. The next is formation of the esters. The alcoholic hydroxyl group of monosaccharides and the reducing disaccharides reacts with the acetylating agents like acetic anhydride in pyridine to form acetate derivatives, acetate propionates, benzoates, phosphates. These are called the esters of their monosaccharides. The sugar phosphates are of great biological importance. Metabolism of the sugar inside the body starts with the phosphorylation. For example, if you memorize the glycolysis, which I think you have studied in your previous classes. So the first step is glucose is phosphorylated into their respective. So glucose is phosphorylated to form its glucose 6-phosphate, and then it's a further reaction, series of reactions get started, and the hydrolysis of glucose started, and the energy produced ultimately. So the phosphorylation have the great biological importance. So glucose 6-phosphate and glucose 1-phosphate are the important intermediates of the glucose metabolism. As I already told you that glucose 6-phosphate is the phosphate form, phosphorylated form of glucose, and which is the first step of the glycolysis and ultimately the metabolism of the energy. Glucose 1-phosphate is another important intermediate of glucose metabolism. Here you can see the structure of glucose 1-phosphate, in which glucose is phosphorylated at first carbon, while glucose 6-phosphate it gets phosphorylated at number 6 carbon. The second important category of the carbohydrate is named as oligosaccharides. Oligo means few and saccharides means sugar. So these are the compounds which contain 2 to 10 monosaccharide units and joined by a specific bond which is called as glycosidic bond. So glycosidic linkage formed via the dehydration reaction. As a result, a hydrogen atom is lost from one monosaccharide and the hydroxyl group from the other, which ultimately combined in the form of a water. So on hydrolysis, oligosaccharides give 2 to 10 molecules of simple sugar units. Further, the oligosaccharides can also be classified as disaccharides, trisaccharides, tetrasaccharides, and pentasaccharides. If we are talking about the disaccharides, these are the oligosaccharides having 2 carbon or sugar units in their structure. The Examples are maltose, lactose, and sucrose. Maltose is formed by the combination of two glucose molecules. Lactose is formed by the combination of one glucose and one galactose, while sucrose is formed from the glucose plus fructose. Next is the trisaccharides. These are also oligosaccharides having three sugar units. The example is raffinose, and raffinose is formed from the combination of glucose, fructose, and galactose. So they have glucose, fructose, and galactose in their structure, and they are combined by a glycosidic bond, a glycosidic linkage. The next is a tetrasaccharide. It is a 4-carbon or 4 sugar units, sugar unit compound, and the example is stachyoses, in which 2 galactose, 1 glucose, and 1 fructose are combined by the glycosidic linkage. Pentasaccharides. The example is verbascose, in which 3 galactose, 1 glucose, and 1 fructose are combined by a glycosidic linkage and form the verbascose, which is a 5 sugar unit compound. Among the oligosaccharides, disaccharides are the most common sugars. These consist of 2 monosaccharide units held by a glycosidic linkage. These are also crystalline, water soluble, and sweeter in taste. They subdivide on the basis of the presence of or absence of the free reducing groups. So they can be reducing sugars or the non-reducing sugars. For example, reducing disaccharides have a free aldehyde or ketone group, so they are called reducing sugars. For example, maltose and lactose are the reducing disaccharides. The non-reducing disaccharides are those sugars which have not free aldehyde or ketone groups. So the examples of these disaccharides are sucrose. Sucrose is a disaccharide which do not possess the free aldehyde or ketone groups. The journal formula is CnH2O(N-1) for the disaccharides. The important example of the disaccharide is maltose. So maltose is a sugar which contains 2 moles of or 2 molecules of the glucose in their structure. They linked by the alpha 1,4 glycosidic linkage, which means they formed by the combination of 2 alpha glucoses, in which the carbon number 1 participate in the glycosidic linkage of one sugar, while the 4th carbon get combined in the glycosidic linkage. So the linkage will be in the maltose is alpha 1,4 glycosidic linkage, which is between the first carbon of one carbohydrate and 4th carbon of the second glucose unit will be, and it is one of the reducing disaccharide which has the free functional groups. So they have a free functional group to get attached with the other moieties. So they used naturally in the fermentation reaction of the alcohol and beer manufacturing. So maltose may be present in their alpha or beta forms depending on the configuration at the free anomeric carbon atom. So it is a reducing disaccharide and it forms a petal shaped crystals of the maltose osazone. The next example of the disaccharide is lactose, which is a milk sugar because it is present in the milk. It contains one molecule of galactose and one molecule of glucose in their structure, which are linked by the beta-1,4 glycosidic linkage. It is also reducing disaccharides and hydrolyzed into galactose and glucose by the enzyme lactase in humans and by the beta-D galactosidase in bacteria. It is a source of carbohydrates in the breastfed infants. Here's the structure you can see in this picture as well, in which the one molecule of galactose is combined with the one molecule of glucose component by beta-1,4 glycosidic. So lactose form osazones, which resembles pincushion with pins, or the hedge hog, or the flower of touch-me-not plant. Here you can see the representation or the figure of the hedgehog or the pincushion with the pins, or the flower of the touch-me-not plant, which is a lactosozones. The next example of disaccharide is sucrose. It is commonly used as a table sugar and contribute some calories in the diet. It contains one molecule of glucose and one molecule of fructose in their structure that are linked by alpha 1,2 glycosidic linkage, which means the first carbon of alpha D glucose get combined with the carbon number 2 of a fructose and form a alpha 1,2 glycosidic linkage by the removal of water. It is a non-reducing disaccharides, which means it is not possessing the free aldose or ketose group in their structure. So it hydrolyzed into glucose and fructose by the enzyme sucrase, which is also called as invertase. Here you can also see the structure of sucrose, which is formed by the glucose and fructose molecules which is linked by the alpha 1,2 glycosidic linkage. On hydrolysis of sucrose, it will produce one molecule of glucose and one molecule of fructose. Therefore, the product will change the dextrorotation to the lever rotation. As we know that glucose is a dextrorotatory substance, while the fructose is a lever rotatory compound. So, therefore, the product will change the dextrorotation to the lever rotation, and equimolecular mixture of the glucose and fructose thus formed is called as a invert sugar. So the enzyme producing hydrolysis of the sucrose is called as invertase. Honey contains invert sugar, which is the combination of glucose and fructose, and therefore it is more sweeter than the only sucrose. Polysaccharides. The third important category of the carbohydrates is polysaccharides. So they contain more than 10 sugar units in their structure. If a compound contains more than 10 sugar units or monosaccharides in their structure, they are called as polysaccharides. They have high molecular weights and are sparingly soluble in cold water. So they are called as glycans. They may be either linear or branched in their structure. They are not sweetish in taste and do not exhibit any of the properties of aldehyde or ketone groups. The general formula for polysaccharides is C6H10O5 (N). Polysaccharides are further divided into two groups. The first one is homopolysaccharides. The second is heteropolysaccharides. So homopolysaccharides means, homo means same. The polysaccharides in which same type of monosaccharides are linked, these are called homopolysaccharides. For example, starch and glycogen, inulin cellulose, dextrin, dextrains are the examples of homopolysaccharides. All these compounds are formed by the same type of monosaccharides in their structure. Heteropolysaccharides or heteroglycans. These are the polymers of different type of monosaccharide units or their derivatives. For example, mucopolysaccharides or glucosaminoglycans. These are the examples of heteropolysaccharides. Here you can see the structure or the conformation of the homopolysaccharides and the heteropolysaccharides. You can see that the homopolysaccharides are formed. These may be branched or unbranched, but formed with the help of same type of sugar units. In case of unbranched, these are the linear chain of the polysaccharides, while in case of branched, they have a branching on a certain number of carbon units or sugar units. So, but they consist of similar type of carbohydrate units. But if we talk about the heteropolysaccharides, they are the combination of different type of monosaccharides. In case of homopolysaccharides, starch and glycogen are the examples of homopolysaccharides in which the same type of carbohydrate, which is a glucose, is a repeating unit of monosaccharide. While in case of heteropolysaccharides, two monomer types unbranched, means two different type of monomers are present in the linear structure of a heteropolysaccharides. The second, they may be branched but have the repeating units of different type of monosaccharides are present in their structure. Here you can see the different colors of different type of monosaccharides are present in the heteropolymers. Either they are the branched or unbranched, it doesn't matter. But different type of monomers are present in their structure. Polysaccharides can also be classified on the basis of their functional aspect. For example, they can be grouped under two heads. The first one is nutrient or digestible polysaccharides. The second one is structural or indigestible polysaccharides. The first one, which we are going to discuss, is nutrient or digestible polysaccharides. These are the polysaccharides those act as a metabolic reserve of the monosaccharides, which are the repeating units of their structures, and both in the plants and the animals. For example, starch, glycogen and inulin are the examples of nutrient or digestible polysaccharides. Whenever the living system encounters the energy deficiency, these reserve polysaccharides get hydrolyzed and liberate the free monosaccharides or simple sugars. These simple sugars further hydrolyzed to generate the energy. For example, starch is the energy reserve or storage form of energy in the plants, while glycogen, which is present in the animals, hydrolyzed into their simpler monomers, which are the glucose, because they are formed from the glucose as a repeating unit in their structure. So this glucose get hydrolyzed to generate the energy whenever the body required. The next one is structural or indigestible polysaccharides. These polysaccharides serve as a rigid mechanical structures in the plant and animals. For example, cell wall's main structural component is cellulose, and cellulose is a structural or indigestible polysaccharide. We know that cell wall functions is to provide the stability and support to the plant cell. Also, the pectin is the structural component of the cell wall and also the member of structural or indigestible polysaccharide category. chitin, which form the exoskeleton of many insects, also provide the stability and support to those organisms, living organisms, and are the structural polysaccharides. In the same way, hyaluronic and chondroitins are also the structural or indigestible polysaccharides. We are not going to discuss the details of the examples of these type of polysaccharides because we will discuss these in our further lectures. So till now we discussed all about the carbohydrates, their definition or their classification in detail. Dear students, thank you so much for having time and listening me. You can also consult these topics from these books. Uh, Hopefully, it will help you a lot. You can also ask me the questions as well. And till now, it's all about what we have discussed is stay safe and do well. Thank you so much. Allah Hafiz.